go to Lecture 1 to start the lecturing now, but there is a website, this one that's shown here, that I'll be clicking back and forth onto as we now begin mostly the verbal discussion of the milestones in development, and then come here to take a quick look at a picture. So this website is what I'll do so it can be more easily read. I'll go to the PowerPoint, Control Home. No. I'll insert a new slide. I'll paste it here, and we'll start the show. So this is the web page that will be supplement to Lecture 1. If you want to enter that in, get that loaded in your Internet Explorer, in addition to having Lecture 1 posted from WebCT. If you don't have access to WebCT yet, it's all right. Mostly what we're going to do today is just kind of cruise through some of the major milestones, and it is primarily reading, and we'll pick up where we leave off at the lecture on Wednesday. So that's www.visembryo.com/baby/hp.html. Let me get my pointer set on arrow. So there's a tremendous amount of terminology that will be presented in this class, but there are also very key concepts that we need to establish. One is for curiosity, because we all develop this way. We all are involved with families being generated and created, or having our own babies ourselves and so forth. So there's that interest. But then in addition, it's expected that, or not an expectation, but typically most of the people that take this class are biology majors. If you get a degree in biology, then you basically have the obligation in the general public to be able to inform people when they inquire about things. And with stem cell research and abortion and all the key issues, in vitro fertilization, cloning, when all these topics are of really key concern in society, then you have the obligation of sharing your knowledge to help clarify things with people. So a good, strong background in this area is essential to meeting that commitment. So there are two major developmental periods: the prenatal, figures 1.1 and 1.2 from the text, which is fertilization to birth. And then there is the postnatal, and that's birth to development. So developmental biology really is the study from conception to death. It's the complete spectrum of development, from the moment we're conceived to the moment that the life force leaves the living organism. Embryology, of course, really is more limited in its studies, and it's the prenatal portion that we are going to investigate. So the human reproduction and development really has a lot to do with the evolutionary success of humans, and in part that's related to the internal fertilization and the uterine or intrauterine development of the embryo. There is the placental trait. So are humans viviparous or oviparous? What about the shark? The kids and I would go through this every time we'd go through the shark thing at SeaWorld. They are oviviparous. They hatch eggs, but the eggs hatch inside, and then they give birth to live animals. So humans are viviparous, and there is intrauterine development, of course, thereby. But we do have a unique kind of placenta as well, that has a hemochorion. There is the open blood lakes in the human uterus, the endometrium, with the implantation of the embryo. In some species, there's placenta, but not the hemochorion. There isn't the opening of the vasculature. So these are some of the key things that we keep in mind about commonalities among mammalian species and uniquenesses of the human situation. The postnatal nutrition of the newborn is via the maternal lactation, a mammalian trait, of course. The development of the limbic brain is key for the nurturing because our young are not precocious. They require a tremendous amount of nurturing. And then the development of homeostatic mechanisms, the avian and mammalian traits that are shared. So there's a significant relationship between embryology and the evolutionarily successful human species. The human development then follows this line of events. There is gametogenesis, the formation of the haploid gametes, the sperm and the eggs. There is fertilization with the fusion of the sperm and the egg to form the zygote. So there is a term for the outcome of conception up until a very key point, and it is the zygote. So we will use specific terminology. We won't use the term embryo until we reach a certain developmental stage that morphologically satisfies the criteria in the definition of embryo. As I pointed out earlier, I asked Jennifer if she's 8 weeks. If she isn't 8 weeks, then we don't refer to the conceptus inside her as the fetus. It is still the embryo. So there's very strict definition of these stages of development. So you may find individuals referring to the embryo erroneously in textbooks or in the literature based upon that strict definition that we'll be using. Cleavage is a special rapid mitotic cell division of the zygote to form the multicellular embryo. So we'll see that there's some unique things about these early cellular developments that we don't always see in other cases of mitotically active cells. And specifically, that is that you'll have cellular division without production of new cytoplasm. So the egg is a very large cell, and it will be split up into equal portions, basically, to produce the first small number of cells. I've forgotten exactly when it is. It's the 8 or 16 cell stage, something like that, before new cytoplasm is going to be developed. So this minimizes the metabolic needs of the cell very early on, and anatomically at that point, of course, we don't have implantation, so the source of nutrients is just the solution that is within the fallopian tube. So these are key things to keep in mind. What limits the development, the rate of development and growth of the conceptus? So when I use this term conceptus, even though we didn't get to all the definitions yet, I'm referring to any stage along the way from fertilization to birth. Whatever there is, the collective group of cells, which is, of course, the chorion, placenta, and all the membranes and the developing organism, whether it's zygote, embryo, or fetus. So that collective embodiment of cells is the conceptus, everything that was produced by conception. So I use that term when I don't want it to be differentiating any particular specific component or part of the developing cells. So another aspect of development is morphogenetic movements. So here we're not talking about genetic like the gene. The term gene originated from genesis, to produce. So here we're talking about the genesis or the production of morphology. So there's cytogenesis is a term that we've used already, the development of the cells. Histogenesis, the development of the tissue. Organogenesis, and development of the organism. So organismogenesis, we stopped using it at about that point. So the development of the morphological, the characteristic morphological appearances occurs really through specialized types of movements. It'll be sheets of cells or clusters of cells that are migrating, or individual cells that are migrating. So there'll be a variety of things that we're going to have to try to envision that are occurring to produce the characteristic feature, three-dimensional structure that we see. In addition, we'll be looking at two-dimensional structures, just sections through the developing conceptus to try to make sense of what's happening. So there's cell and tissue differentiation. So the part of cytogenesis is differentiation. We will talk about mitosis and meiosis, and one of the first things that I would do when I was teaching the Bio 1, and I still bring it up when I teach the AHS Human Anatomy class, the cellular part of it, is that we challenge the idea of the mother cell dividing to produce two identical daughter cells. This is not true at all, because when the mother cell divides to form two daughter cells, one of those really is a mother cell that maintains the full mitotic capacity and replaces the mother as the reserve cell. So really the mother cell gives birth to a daughter cell that and stays as she was, changed somewhat. You'll never be quite the same, no matter what your doctor tells you after you give birth. And the daughter cell will go on to differentiate into the post-mitotic functional cell. So it's very important to understand that when we think about the adult organism, where our skin cells are dying and falling off, the mucosal cells are dying and falling off, our gastrointestinal lining virtually is replaced within every 4 to 6 days. So we can understand it in that sense. The deep layer of cells are the reserve cells, and they're giving birth to daughter cells that differentiate into the post-mitotic functional cell that lives for just a certain period of time, dies, and goes away. But what we're having with the development of the organism is mitotic division, where there's not a tremendous amount of cell loss. We're just dividing and producing and dividing and producing, and we start with what is called the totipotent cell, a cell that has the potential of becoming anything. So the true stem cell that is totipotent can become anything. Then there are other stem cells along the way that have limited routes of differentiation, so they might be pluripotent. So one progenitor cell that can become all the different types of connective tissue cells, for example, and produce all those lineages, or they might be able to produce all the lineages, the stem cells for producing all the different blood cells, the red blood cell, the different kind of white blood cells. So when we hear the term stem cell out in the public, everybody's thinking that an embryo or a fetus has to die to produce that, but it isn't true. The stem cell is a term that has many, many different definitions, if you will, depending upon how you're looking at the development. So we'll make an example before we come back to this idea of what's going on in the development of the conceptus. Saw an article about a dentist that just happened to collect one of his kid's teeth when it came out, took it right to the lab and put it on ice, took it to the lab, and washed out all the little cells inside it and isolated stem cells from that could differentiate into connective tissue cells, teeth-forming cells, nerve cells, the nerve protective cells, the myelin cells, and things like that. So those are stem cells. But no embryo or no fetus had to die. A kid just had to lose their tooth like they lose all the time. So it's important that as biologists we understand that, because most everybody in the general public, when they hear the word stem cell, all they can think of is abortion. So it's important that we understand that. So we have these cellular lineages of differentiation. The totipotent cell that can become anything. To get that, you either have to de-differentiate a pluripotent cell, which we're not really very good at understanding yet, or you have to collect it or procure it from a very early conceptus. It may not be from the embryo or the fetus. It might have to be from the umbilical cord or something like that. So we need to understand that. So what it means for us is that at any one point in time, when we're following cytogenesis, histogenesis, and organogenesis, we're going to see cells of different potency within a little spot on a histological slide that we might be looking at. They might be multipotent or totipotent or pluripotent, some type of term like that, or bipotent or unipotent. So there's certain levels of commitment as we go along. So this cell and tissue differentiation is a concept when we see that set of terms, which should invoke within us this whole picture of this broad spectrum of cells, many of which can be called stem cells, and they're either predetermined to a very limited degree or a not so limited degree as to what they can differentiate into. So this is where we start taking words and putting these concepts and pictures in our mind. Right now we should be seeing one cell, making several cells, making several cells, making several cells, breaking out into lineages that can become a neuron, that can become an adrenal cortical cell, that can become a skeletal muscle cell, that can become an osteoblast, and so forth. So we will see. So now I mentioned the term osteoblast. What does an osteoblast get called once it has become the mature, fully differentiated post-mitotic cell? An osteocyte. So we'll hear this term blast. So a blast cell is a type of a stem cell, but it's typically unipotent, especially in the example that we just gave. So we need this type of thinking in biology because we don't always have to kill an embryo or destroy an embryo. We're not always killing it. We're harvesting cells. We're disassembling an embryo, I guess, in order to get stem cells. Once we understand that full definition of stem cell, then we know there's more places to go and look for cells that are already predetermined. So if we want to produce a particular cell line or a particular tissue, then we go looking at certain locations to do that. So pattern and polarity then become very important. So we've got these undifferentiated clusters of cells that are just kind of moving and dividing and reorganizing. Somehow they're figuring out where they're supposed to go. We'll see that different cells from different layers have got to end up in certain locations in order to produce certain organs. So it's highly organized. And we just look at it in awe in this class. In the developmental biology, they look at all the molecular aspects of what is known about it. So if you really get taken by that aspect of saying, how do they know where to go and when to do this and when to do that, and how is it determined that they differentiate along this line, you may find yourselves taking the developmental biology in addition to this class to open up new avenues of understanding and investigation along these lines. But there's going to be patterns and polarity. we certainly don't want our foot cells ending up to at the top of our head and growing out of our head. So something knows or something tells cells to go someplace and develop into our toenail versus the hair that grows on our ears once we turn 30 or 40. Didn't have hair growing on these little parts of my ears, the antitragus, when I was in my 20s, but now all of a sudden I find myself plucking my earlobes, not my earlobes, my antitragus just about every week. So I'm glad it's not my toenails that popped up there some 40 years later. So we have to have the complete symmetry of the organism. If things don't happen the way they should, then we end up with potential aberrations. What's an aberration that happened just recently at Wilford Hall? Conjoined twins. So didn't get the complete destination signal sent to the organs. We go to 250, is that correct? So we're just at this point. So growth and maturation are a key part of it, and then as I just indicated, the senescence of my ears, starting to grow hair, and then ultimately the end point for all organisms, death is a part of life. So we'll pick up with this